The neuronal ceroid lipofuscinoses are a group of genetically determined progressive disorders leading to a complex neurological picture, including dementia. All but one are autosomal recessive disease. NCL forms are related to 13 NCL genes, which encode the proteins of the lysosomal compartment, the endoplasmic reticulum, the cellular membrane. Characteristic pathological feature of the NCLs is the endolysosomal storage, which occurs due to either the lacking activity of a specific hydrolase of the lysosomal matrix or to the impaired protein degradation process. Examples of pathological storage are shown in this slide. Phenotypic heterogeneity is quite common among the NCL. Mutations of the same gene can rise to different phenotypes in terms of age at onset and disease duration. Aim of the study was to investigate the lifespan of a cohort of NCL patients according to their genotypes. The phenotype heterogeneity was examined focusing on variant and genotype pathogenicity. A cohort of 152 children selected from the database of the Italian network of NCL was grouped by the age at onset, infantile, late infantile, and juvenile forms, and subdivided according to the associated mutated genes. Selected clinical features, age at onset, disease duration, age at death, were assessed by descriptive statistics and significance level measured by non-parametric tests. The pathogenicity of the individual genotypes was categorized following the guidelines of the American College of Medical Genetics. The median age at onset of the whole cohort was 4 years. The median age at death was 17 years. Statistically significant distinct curves of survival probability were obtained after Kaplan-Meier analysis, as you can see in the bottom sketch of the left column. Meaningful differences of lifespan among groups and distinctive features within each group were detected, as well as high correlation of the clinical parameter, such as onset and age at death, as shown in the mid-column slides. High percentages of pathogenic variants were identified, and biallelic pathogenic variant accounted for about 90% of all genotypes, all equally distributed about the groups. NCL forms associated with mutations of genes coding for lysosomal enzymes at a more severe course, as shown by the violin in the mid portion of this slide. In conclusion, this study has shown that the lifespan correlates with the age at onset only partially with the mutated gene, but not with the pathogenicity scores. Age at onset correlates with age at death and disease duration. The earlier is the age at onset, the faster disease progression, and the earlier age at death, regardless of the mutated gene. Variance pathogenicity and related genotypes do not correlate with the age at onset, nor with the disease course. Severity of phenotypes observed in patients mutated in the same gene were not associated with variant pathogenicity score. Natural history studies from large cohorts, including score genetic variants combined with fluid biomarkers, are recommended to improve the characterization of phenotype heterogeneity. That might help to identify the mechanisms which establish the differential time of onset of the disease phenotypes associated with mutations of the same gene, and investigate the role of variant and genotypes on establishing the phenotypes.